In this video, we interview Dr. Quilici, the director of the Nevada Proteomics Center, to gain a deeper understanding of real-world applications of mass spectrometry. First, we asked what a mass spectrometer does and what can be measured.
Hi, I'm Dave Quilici. I'm the director of the Nevada Proteomics Center. This is a core that analyzes protein using mass spectrometry. Mass spectrometry is the instrument that analyzes and measures ions. And so these ions can be from elements, they can be from molecules, they can be from macromolecules, which a protein is a macromolecule. It allows us to get the mass of that ion and then determine the structure or what that compound or element might be.
How can mass spectrometry be used or applied?
Proteomics, or we use mass spec for proteomics. And mass spec can be used for multiple different applications. It can be used in chemistry, maybe synthetic chemistry, to know exactly what the molecule they made is. And so you can make sure that you synthesize the right molecule. And you can use it, in our case, for proteomics, where we are identifying and quantitating proteins in certain disease states. And this allows us to determine pathways that are affected by a certain disease state, possible modifications to proteins that affect the function of this disease state. Or also, we can do biomarker discovery, where we can actually start looking at a clinical approach for markers for a certain disease state and possibly catch it prior to evolving.
How are samples loaded?
So this system that we have here is an LCMS, and so you might see other systems like be integrated with a GC. So LC is liquid chromatography. So proteins are in a liquid solution that go through a column, and the column is here on the front. And the flow rate is at 350 nanoliters a minute. So just to get a perspective of flow rate and how low that actually is, a drop of water is about 10 microliters. So it takes upwards of 30 minutes to get the 10 microliter drop out of the system. So it's very low flow. Our sample is introduced, goes through this column and separates, because they tend to be very complex. They then go through into the mass spectrometer. This here is an ionization source. So we're going from a liquid to a gas phase. And so our molecules have to go from this liquid medium into the gas phase. And it's kind of hard for a protein to do that because it's quite large. If you think about a small molecule, it's fairly easy, and that's why we use it in C for those. But we have to use these in a liquid chromatography setting to get the iodine. In this case here, to ionize them, we apply around 3,000 kilovolts to ionize them. And this basically gets rid of all the liquid, the buffer solution that's eluting with them, and allows those ions to go into the mass spectrometer.
What are the main components of a mass spectrometer?
There are three parts to a mass spectrometer, three main parts. The first being the ionization source, and this is where we transfer all of our liquid into the gas phase, and we get all of our molecules of interest into an ion. So they have to be charged for us to actually analyze them. And so once we form those ions in the ionization source, they can then be directed to the mass analyzer. The mass analyzer then is able to select them by their mass to charge, and then these can be measured once they hit the detector. And so the instrument is fairly complex in the fact that the mass analyzer has a radio frequency associated with it, and it also has a magnetic field associated with it. And what happens in this quadrupole that's actually in front here is these ions can then cycle through to get to the detector, and only one mass can get through to that detector at any given time. And so this instrument is very fast in that Each ion can go through the mass analyzer in microseconds, but we're looking at thousands and tens of thousands of ions at any given time. So the instrument has to be extremely fast. This is one of the fastest instruments on the market, the Orbitropic Eclipse. And it also has a resolution of 480,000. So we can actually see down to the fourth decimal place.
How are samples read?
So these ions, once they are separated by the mass analyzer, they then traverse to the detector, and they're detected there. One of the interesting facts about this is we are not able to detect a single molecule. And when we're separating these, we need to amplify that signal. So we use an electron multiplier. So when our actual compound strikes the detector, it starts a cascade. And so it amplifies the signal, and so that we can actually see a peak coming off with our molecule coming off, or our ion. Because again, we can't see one molecule, and we're looking at only a few hundred, and we can't even detect that right now. So these molecules strike for our electron multiplier, they start a cascade, and then we can actually see that signal.
How long does a sample take to run?
So we are in a proteomics core, and so our samples tend to be extremely complex. We're looking at tissues, cell lines, and there's tens of thousands of proteins in a given sample. So when we separate by LCMS, we're able to separate all these molecules, all these proteins, so that the instrument has plenty of time to actually analyze it. And to do that for a very complex mixture, it can take up to runs of like 2 hours long. So if you're looking at more clinical approaches, where you're looking at the serum or urine and the proteins in those type of samples, we can do those in 10 or 15 minutes. So it kind of depends on the sample, how long it's going to take to get through. But the key is you have to have your samples spaced out enough so the instrument has time to analyze each and every molecule that's coming through.
What are some challenges of running a mass spectrometer?
So LCMS is a very complex instrument. You can see that here. We're running at very low flow rates. This is probably where all our failures happen, is mostly in LC. And that's because we have to have connections that are very snug, and we can't lose any liquid when you're filling at only 350 nanoliters a minute. Even the smallest amount of liquid changes or flows through the system and our reproducibility. So the LC is really one of the factors, that and having spray. Having our molecules come out with a nice steady spray, and that's going from the liquid phase to the gas phase. That has to be very consistent. It can't be bubbling, it can't be sputtering in any way. So that has to be a very consistent spray. The instrument itself, very complex, so there's always an issue that could happen there. We're dealing with liquid coming through, we're dealing with 3 to 4 kilovolts in this place right here. And so we always have some issues here, but typically it's the LC that prevents any.
How are results seen and interpreted?
So once we get our results, this is what they will look like. On this top panel here, this is the chromatogram. And so this is the elution of all of our ions off of the LC system as they come off. And at any given point, as you can see right here at this 56.37, we have this ion 584 coming off, and you can see its isotopic cluster there. We also collect MS2, which would be the fragmentation of that, so that we could find the amino acid sequence of that 584 ion. And all the way across this chromatogram, we're collecting these data points throughout, and we will collect somewhere in the order of 50,000 data points, which means we have 50,000 MS1 and MS2 for us to process further on down to determine the pathways and how these proteins are changing in a given disease state.
Thank you all for watching. I hope you enjoyed this interview. This video was created by me, Gabriella Goodwin, as a member of Dr. Ryu's computational lab group. Special thank you to Dr. Quilici and the Nevada Proteomics Center, as well as Dr. Ryu and the members of the Ryu Computational Lab Group. Finally, I'd like to thank the NIH as well as the NSF for funding this research.